biodiversity, there has been a discussion on the importance of open science, open data, on the capacity of sharing and using, capitalizing the data. And actually, it is one of the most relevant issues we need to face now, because it is absolutely impossible to address the challenges that we have in front of us with all the potential threats to biodiversity and ecosystem, including climate change and global warming as one of the most relevant, if we don't manage to capitalize on all the data that are collected by the scientific community, that are collected by the different agency and administration, and build on that. So giving the possibility to share, use, reuse the data, and then build on what has been already built by other scientists. So that, I think, is basically the philosophy of the project that some of you are leading, because I imagine that there are going to be a coordinator of the Biomon project that are being funded by the Biodiversa Plus call. I think it's something that is extremely useful and important to us. There are, of course, we could say, not critical point, but delicate issues that need to be addressed, particularly for the data collectors, the data providers that are spending months of their time to collect the data, and then surely want to use the data before sharing. But sharing is something that is a great success for all of us, and then we have the possibility to recognize the work done by the data collectors and data provider. We have the possibility to give the DOI to the data set. We have the possibility to have data published in terms of data papers. So there are a lot of efforts that are supporting the process of linking data collection with data utilization at the broader scale. And then in the middle, there's a lot of technology. And then we are going to discuss today not only with the need or with the relevance and with the components for the achievement of a complete fairness and openness of data and data sharing, but also on the technology that can help us in order to facilitate this step, facilitate all the step in the data curation, facilitate the capitalization on the existing data. And that is basically what we would like to discuss today, having this quite short workshop divided into components. A very first one, which is more focusing on data fairness and data harmonization and technology that can support data harmonization. And the second half of the meeting, which is more focusing on one of the most widely used and most fast-growing standard for biodiversity data, which is the Darwin Core standard. Darwin Core standard is applied as different potential way of application, is applied in different way in different countries and in organizations. That's why we have a number of presentations of that. So I will not take longer, since we have a quite dense agenda, and therefore I will opening the workshop, and I will start giving the floor to the first presenter that are going to discuss on making research data fair and usable, which is also going to be an overview of metadata harmonization. I'm giving the floor to Senem, Tarantini, and Martina Pulieri, that I'm also thanking them so much for supporting and joining me in the coordination of the task 2.2.2 of Biodiversa Plus and the organization of this workshop. So the floor is yours, Senem and Martina.
I think we can start with the Menti. Okay, so we have some mixed answers here.
Yeah. Mostly we know how to organize our data set. It's nice.
It's perfect. Okay, so I think we can start.
Yes.
So Senem, the floor is yours.
Okay, thanks. Good morning, everyone. My name is Senem and Tarantini, and along with Martina Pulieri, we are researchers at the University of Salento, Italy. We are working with Professor Alberto Basset on the Biodiversa plus subtask on harmonization and interoperability, and we also work for LifeWatch Italy. Today, I will pass through general description about harmonization and fairness. Then our invited speakers will go in detail about harmonization process, tools for harmonization and fairness. We are repeating FAIR principles, and today you will hear many times FAIR principles and fairness. FAIR is acronym of findable, accessible, interoperable, and reusable. This isn't just a set of letters, it's a guiding principle for ensuring our biodiversity research data is truly useful. Findable means that it should be discoverable through clear and detailed metadata. It should register to the public repositories, which have search functionalities, such as data could be done by taxon-based, area-based, habitat-based. It makes easy data discovery for both human researchers and machine. Accessibility, we should provide clear instruction for accessing and downloading the data, and we should use open data formats whenever possible. Also providing long-term data storage in trustable repositories makes data accessible. And interoperable, biodiversity data comes from diverse sources with many formats and methods. Standardized vocabularies, units, and data structure ensure seamless integration and composition. And reusable, making data usable for future research. How we can do it using standard methods and units, free access data license, and these things help researchers to build upon existing data and accelerate scientific discovery. Harmonization process go on with two levels, and data harmonization deals with directly dataset, and the aim is organizing dataset in standard structure. Data harmonization is organizing information about dataset in a standard structure. As you can see, biodiversity data is quite fragmented. Similar information comes from different sources and in different forms, like remote sensing, in situ observations, citizen sites, museum records, etc. Like imagine you are in a library full of books in various languages and writing styles for the same subject, and you don't have any idea how to start to searching what you need. With the harmonization process, we bring order to this chaos by identifying formats, resolving inconsistencies, and giving a standard structure for all datasets. Like you find a translation of each book and the information in the same structure. Okay, this is a quick and real example. This was one of the worst cases because the data set in PDF format in Italian and crucial details like sampling protocols, geographical coverage were lost in the text. There were limited taxonomic information. Local names of organisms was given without describing what they are. Here we see dataset after the data harmonization. Dataset has been translated to English. Geographical data becomes detailed from country to minimum geographical area. Taxonomic information is given in standardized form from phyla to species level, and sampling methods and the sampling area findings are clearly explained in datasets. Before, if you are not an Italian researcher, this information was impossible to reach. Now, researchers from anywhere can access, understand, and utilize this information. This is also a great example how data sharing and data harmonization bring value to your data and your efforts. Okay, as I mentioned before, while data harmonization focus on data itself, metadata harmonization focus to descriptive information about data. With the metadata harmonization, information about the dataset becomes consistent across different sources. How we can do this? By using standard vocabularies. Darwin Core is widely used vocabulary for sharing information on biodiversity. We can share standard metadata structure. We can share definition for describing the metadata. This is the metadata of previous dataset. Here we are able to reach some essential elements like dataset name, source of reference, time coverage, who's data provider, geographical coverage, taxonomic information, information about who performed the harmonization, if usage of data has limitation or not, sampling methods, etc. This info not only helps to making data fair, also gives the possibility to researchers, if dataset fits their needs, without going into dataset. Data harmonization is a powerful tool for empowering biodiversity research. By ensuring fairness, we give possibility to the researchers worldwide to collaborate effectively, leading to a deeper understanding of our planet's biodiversity. I will briefly touch to interoperability. Interoperable data means it can be integrated with different datasets, application, and workflows. There are different layers of interoperability as organizational, legal, technical, and semantic interoperability. For the semantic interoperability, I give floor to Martina.
So, yes, as Senem was saying, data interoperability is quite a challenge. But data interoperability implies also semantic interoperability. And as defined by the IOSC interoperability framework, semantic interoperability ensures that the precise format and meaning of the exchange data and information are preserved and understood throughout the exchanges between parties, and in this case could be both human and machines. Always from the IOSC interoperability framework, they set a few problems with the semantic interoperability. A few of them I reported here. The first one being the lack of common explicit definition. So we have different vocabularies, and among these different vocabularies, there are different definitions about the same concept. Then there is also a lack of common semantic artifacts across communities. In recent studies carried out by our group, we found that 540 semantic artifacts about biodiversity and ecosystem studies were published. And we found numerous overlapping in topics. And most of them were created ad hoc for projects and never mapped to the existing semantic artifacts, in fact, impeding interoperability. And this lack, and there is also a general lack of expertise and skill related to semantics, which this will negatively influence the use of common definitions. But the IOSC semantic interoperability framework gives us also some recommendation, which is going to be the last slide from our side. So we have to agree on a clear and precise definition for concept to use for our to describe our data. So the use of commonly agreed vocabularies to allow an easier also exchange of data and data integration. And also a really important part is to thoroughly document your data. So to describe the what, who, where, when, how, in order not to lose information and to allow the reuse of data.
Thank you, Martina. Thanks, Senem, for this introductions on the basic components of the fairness and harmonization. I'm really happy to give the floor to Naouel Karam, who was so kind to accept our invitation, and then is going to give a presentation on the semantic technology and their usage for biodiversity data. So considering both opportunities and challenges. So Naouel, the floor is yours.
Thank you for the invitation and for the opportunity to present our work. So I'm Naouel Karam. I'm from the Institute for Applied Informatics from the University of Leipzig. And I will present our work in the context of NFDI for Biodiversity, a German project. So I structured my presentation in 3 parts. So the why do we need semantic technologies? Then what are terminologies? What do we mean with terminologies, and what terminologies do we need? And how do we use terminologies to enable semantic search and semantic interoperability of biodiversity data? So I will start with the why. So the main reason is the ambiguity of natural language. So we have different words for expressing the same concept, which is known as synonymy. If I search, for example, for this beautiful butterfly, Adonis blue, using the scientific name, I can also search for the same concept with using a synonym, or no, sorry, this is, I think, the common name, and then using the synonym. So basically, all this information needs to be stored somewhere, and this is what, in this example, in taxonomies like the NCBI taxonomy. So you can see that the information about this concept is here, like the scientific name, the common name is here, Adonis blue, and the synonym as well, and additional information that is coming from the taxonomical hierarchy and so on. So the second issue or about natural language is called polysemy, which means that we are using the same words for different concepts. An example would be with mercury. If I search for mercury, you can see that we have results for mercury, the planet, and mercury, the element. So the same this information can be found in what is called terminology, or in this case, the ontology GBIF, so the chemical element for biological interest. And you can see, for example, here for mercury, saying that it is the element mercury that is meant here, and additional information is also available, like saying it is a neurotoxin, a poison that interferes in the functions of nervous system and so on. So additional information is also available in ontologies. So this means we need to talk about keyword concepts instead of talking about keywords. And you can see it in Google does it since many years, this differentiation between concepts. Like, for example, if I start typing apis, then we can see there is, the first one is the honeybee, the second one is also an insect, and the third one is a plant. So there is already differentiation what is meant behind. So if I click on honeybee, then we see that this information is already stored in the knowledge graph from Google with the scientific name, lifespan, and some additional information also about the lower classification, the class, family, order, and so on. So all the taxonomical information is already taken into consideration during the search. So this is what we mean with semantic search. So this means, what Martina said, we need to have meaning to express which can be expressed using terminologies. So what do we mean with the terminology? It's also called either semantic artifact or semantic resource. So if we go from the most informal to the most formal, we can go from controlled vocabulary until an ontology. So a controlled vocabulary is a simple list of terms. A glossary is a list of terms with additional definitions of the meaning of the words. For example, our GFI record base is a glossary, which is basically a list of terms with their definition. A taxonomy, so I think this is well known. It's a list of terms organized in a hierarchical structure. An example is from our partner project AMOD with this IOD bird list. And you can see that the terms are organized into structural hierarchy. Then we have thesaurus, which is a controlled vocabulary connected via relations between terms. An example of such relation would be narrower border, so hierarchical information, related terms that connect terms at different levels, and synonymy. An example of such thesaurus would be the Eurovac thesaurus, and you can see that biodiversity is under biosphere, and it is related to animal resources, plant resources, protected species, and so on. And finally, the most formal kind of terminology or semantic artifacts are ontologies, which are more formal and basically based on logical representation and logical actions. An example would be the KB ontology I showed earlier, where Mercury is represented within the hierarchy, with the synonymy, but also more complex descriptions saying, for example, that it has a role as a neurotoxin. So this is what we mean with terminologies, and to see what kind of terminologies we need, both for our use cases. For example, the first one would be semantic search. We did some years ago work on what biodiversity scholars search for. So we collected a set of questions biodiversity scholars would have. For example, do butterflies occur in calcareous grassland? Is there an influence of geographical elevation on the growth of this species, and so on? So, and then we detected what kind of concepts are used in those questions or topics, and we organized them into high-level entities for biodiversity, which are, of course, organisms, but also terms about the environment, about processes, methods, questions about location, of course, about quality, phenotypes, entities, and also chemical substances or materials. And so then we looked at the coverage of those concepts by ontologies or terminologies that are available. And for instance, for organism, we could use the Catalogue of Life, the DNT by taxonomy, ETIS, and so on. For environment process methods, there is many concepts that are available in the environment ontology or the suite ontology. For material and substance, we are using KB. And then we have many phenotypical ontologies, which are like Pato, Flopo, and Toptisaurus. And finally, for location, we are using geonames. So then I will talk about terminologies for metadata harmonization. We are mainly using two standards for that. The first one is the ABCD, which is specifically for collection data. And you can see here the term dataset with its properties. So we are covering such kind of properties for datasets like the title, the taxonomical term that is covered, or creators, and so on. And this standards is also defined as an ontology, so we can have a semantic representation of our metadata. So I can here show you an example from the Botanical Garden in Berlin, where they use ABCD to describe the metadata of their datasets. Then we use also schema.org to represent metadata and also to add it to the web pages of the datasets, and so that can be found by Google and added to the Google search, dataset search. An example would be from our partner Pangea, and you can see here the representation of the dataset with the UI saying it is type datasets, giving the URL, and also connecting it to creators with their ORCID, for example, and so on. So the information is semantically represented. And the final use case, that is data integration. So we've seen in the first presentation that it's a huge problem to integrate data because it's not standardized. The structures are different. The tables are different, and so on. So I have an example here from the TRI data bank, which is the plant trade database, and you can see. So what we need is to achieve first structural interoperability, but also semantic interoperability, which means that we need to have a standard for the structure. And for that, we use, in this specific use case, the ecological traits data standard that have been defined by our colleagues, and which says, for example, that a dataset needs to have a trait ID and a trait name. So using standard trait names, using standard species names, and so on, like here in this example. And to achieve the content, from the content point of view, we also need to use standard vocabularies. And in this case, specifically, we are using Catalogue of Life for species names, we are using the top thesaurus for trade names, and we are using the units ontology QDT for the units. So to have also standard content in addition to standard structure. And once we have that, we can transform basically this data to a semantic format. I show you an example from the trait database where we transform it into ADF, and you can see, for example, all the red points are trait data, and the blue ones are the identified taxons in those data, and so on. So we have all the connections in the knowledge graph. And this can be queried, of course, and through semantic web standards. Okay, I see that it's already 15 minutes, but I still have some additional slides. I will try to go quickly over them if it's fine.
This is fine.
Okay.
Well, we are in advance, so you can take your time.
Perfect. Okay, so how do we use those terminologies to achieve all this semantic interoperability? So we have our goal is to develop semantic standards, services, and tools for the discovery, integration, transformation, and harmonization of heterogeneous data. And we do that since over 10 years now. So I will tell you about a little bit of history of our project. We started with the German Federation for Biological Data project in 2014, and we already had our terminology service up and running in 2015. Then the NFTI for Biodiversity project started in 2020. So it is the National Research Data Infrastructure in Germany, and it involves many partners and many consortia. So we have 26 consortia already for many domains, like, for example, NFTI for chem, for earth, and so on. So we are involved here, NFTI for biodiversity, and for many domains that are own consortia. And in 2022, we joined the Onto Portal Alliance. You can, I think many of you are familiar with EcoPortal. So this is ontology repositories in different domains that are based, that are working together and developing this code, which is called Onto Portal, that is based on the historical repository BioPortal. And our repository is called BioDiv Portal, and it's up and running since last year. And this is the URL for it. So I go quickly on over the requirements. So we defined a set of requirements for what we call the terminology service or an ontology repository. And basically, the first requirement is that we want to store, search, browse, and visualize the terminologies, which we can do on BioDiv Portal on the Browse tab. The community can contribute with their own terminologies by submitting ontology to the portal. Then we need to offer efficient semantic annotation for text and tabular data. For that, we can use this annotator service where we can give text and then get annotated with concepts coming from the ontologies we have on the portal. Then we want to generate automatic mappings between terminologies to achieve the interoperability. This can be also done. And this, I show you here an example between the Catalog of Life and the ETIS, Integrated Taxonomic Information System, where the mappings are automatically created. Then we would like to provide an API, of course, to be able to programmatically access all the services. And an example of one of our partners that developed a search drop-down menu where we can just type in. Okay, so I will get back to the slide with the API. So the calls are available, and like we can programmatically access all the services that are available in BioDiv portal. And an example is like a developed search from one of our partners. They integrated our API in their web search for terms from taxonomy. And so that's a drop-down menu where you can see, for example, the information about the terms that are coming from, in this case, the ETIS taxonomy, the hierarchy, the definition, and so on. So that the users can decide which concept is best for the description of their data. So I will finish with our ongoing work. The first one is that we are collaborating with our partners from the Portugal Alliance in order to cover additional requirements. So basically, those requirements are only partially covered by BioDiv portal, and the first one is that we want to offer an environment for the development, creation, and publication for terminologies. Like for Workbench, for example, would be a solution, or Web Protégé, and so on. Then we would like to provide efficient versioning and evolution mechanisms to know what happened between versions of terminologies. And finally, we would like to improve the community feedback mechanisms to connect to our users. Another ongoing work is to combine large language models like ChatGPT with semantic technologies, specifically for improving the automatic text and tabular annotation, and also for terminology enrichment in case of emerging vocabulary, for example, that is still not defined in the ontologies or terminologies. Okay, that's it. Thank you for your attention, and I would be happy if there are questions.
Thank you so much, Naouel, for your inspiring presentation, and also for this opportunity that you are launching also with your last slide of new technology and potential useful application of this new technology. We have Menti, which is open.
There is an hand raised by Yiming. So please go ahead, Yiming.
Yeah, hi. Thank you very much for the presentation. My question is related to, because this workshop is supposed to be about Darwin Core, and I think I'm a little bit confused between the ecological trait data standard and the Darwin Core standard, and what's the relationship between them, and when should we use ecological trait data standard and when to use Darwin Core standard? Can you please like the new?
Yeah.
Thank you.
So yeah, basically, I think both standards are meant to describe metadata and data level, and it's a matter of choice. So basically, our use cases call for a different standard. So I mentioned the ABCD standard, which is also a standard that enables both. So basically, there are many of them, and the choice of which standard is best for use cases depends on the data, depends on historical questions and so on. So yeah, I think there is no best standard, but best standards for your own use case. And there are also many crosswalks between those standards. So they are already mapped. So that if there are data that are described using Darwin Core, ABCD could be mapped or transformed into the other standards. So so that would be my answer. We are using, as I mentioned, for trade data, the ecological trade standard, because it's very specific to this kind of data, and the other ones are like more general, and the ABCD is more for collection data. And so I don't know for the Darwin Core, because we don't use it in the project, but maybe someone else could answer this for the Darwin Core part. But yeah, anyway, there are crosswalks, and the mappings also of the terminologies would help to enable the interoperability between the standards.
Excellent. Thank you so much. I appreciate it.
Thank you.
Okay. We have also another question in the chat. What is the best way to add ontology to an existing database?
Yeah, so an existing database means like in an interface or a user interface where data is shared. So as I showed this drop-down menu where users can type enter their taxons, for example, and this is automatically connected to our services. So basically, to connect those two, there is a need for either a widget or an interface for the users that connects to our services. So there is, let's say, something in between the user and the terminology services. So there is a need to develop such kind of interfaces to be able to do that. I hope this answers your question.
Okay, it makes
So,
yes.
can I?
Yes.
Yeah, thanks. Yeah, but I have million of records, so I cannot do by hand. I should find a way to automatically connect my data with the ontology.
Yeah, okay. So you mean this is like, for example, what I showed with this annotation service, where like a text can be sent to the terminology service, and you get back like automatically annotated part in the text. Or it could be also done with tabular data. So basically, the information inside the table, like the names, for example, of the species, could be automatically mapped. So this annotation is what can be done automatically, and so you connect directly the URIs, the identifiers from the taxonomies or from the ontologies to your text, basically, and
okay,
and your data. Yeah.
And later I should add this ID on my database.
Exactly. So this is very important.
For all the column that I want to map with an ontology, I should add the ontology ID.
Exactly. Okay. And this is a prerequisite, let's say, for interoperability, and yeah.
Could I?
Thank you so much. I would suggest. Yes, I would suggest Luca, to keep in touch with Naouel eventually through mail, or we send us the question. We are going to distribute the question to the speaker, because we are a bit late with the schedule, and then we would like to avoid that participants are going to enter in order to attend specific presentation, and then they don't manage to find out because we are a bit late. So, but thank you
Yeah,
for
sure. No problem.
for
Thank you.
both in the discussion, Luca.
Yeah,
you so
thank
much.
you, Luca. Yeah, we can keep in
Thanks so
touch.
much, Naouel.
Yeah, thank you.
Thank you so much. And then I will give the floor to Ilaria Rosati to discuss about the approaches for an open and fair research life cycle.
Thank you, Alberto. So I am Ilaria Rosati from National Research Council, and the aim of my presentation is to provide you main information about the open and fair research life cycle. According to the UNESCO recommendation, open science is a set of principles and best practice that aim to make scientific research for all kind of domain accessible to everyone for the benefits of scientists and also society. The recommendation aims to ensure not only that knowledge, scientific knowledge is accessible, but also that the production of this knowledge itself is inclusive and sustainable. Open science builds on key pillars like open dialogue with other knowledge system, open communication system and social actors, open science infrastructure, and in the last, the open scientific knowledge. About open scientific knowledge, it refers to open access not only of scientific papers, but also data, metadata, educational resources, software, code, and hardware. And one of the most important things in open research is related also with FAIR principles that are published in 2016. In order to not bore more you, because in the previous presentation, but then also yesterday, the FAIR principles are described in detail. I want only to remember that FAIR is not the equivalent of open, but open needs to be fair to be useful. What mean that if we make our data, but in general, all digital outputs of our project openly available, this is not mean that are usable. So in order to be usable, this digital object need to have clear and detailed information and description. Data and in general digital resources can be fair but not open, and the maxim about fair is as open as possible, as closed as necessary. So ideally, we want to have fair data and digital resources and open. So why is important the open science and FAIR principle in general? Because they provide us different benefits at economic benefits, but also because give us the possibility to have global collaboration, to open all our research output to different research community, and also to reduce the duplication in scientific efforts. Despite the scientific community is increasingly recognize the value of open, reproducible, and transparent research practice, a lot of researchers fail to share their data or in general the output of their project because they have different barriers. Some of these are related with the knowledge barriers, for example, because researchers have difficulties about how to archive or to manage data or other research output, or they have concern about, for example, the reuse of their products, and at the end they have misaligned career incentive. Moreover, one of the main barriers that researchers have is related with the lack of time, because the management and of data, or in general all products of our project, is time consuming. So the researcher need a lot of time to prepare, to clean, to manage the data, the model that they produce. But in order to support this process, there are institutions in general, but also research infrastructure that can provide not only tools and services that can facilitate the process, but also provide data management teams that can work together with the researchers. About this, I would like to show you an example of approach to open fair research life cycle, adopted and constantly evolving in LifeWatch Italy, that is one of the national data distributed center of LifeWatch ERIC, the research infrastructure on biodiversity and ecosystems. This life cycle includes different steps, from planification to reuse of different research products. So I will focus not only on data, because during your project, also in your project in Biodiversa Plus, you produce also model, algorithm, or software code, different codes. So it's important that all these products are open and fair in order to be, after the project, reusable by other researchers. So the first step is related with the data management plan, and the data management plan is very important for producing fair output. For this step, LifeWatch Italia rely on external resources such as the Argos tool that is provided by OpenAir. This tool creates machine actionable data management plan. Moreover, provide different templates. Some of these templates are aligned with the Horizon Europe project. There is a link with the European Open Science Cloud. So we select this tool because as we're at different our requirements. But one of our idea is in the future to directly integrate this tool, or we are also analyzing this moment with a tool in order to integrate this tool in our management system. In this way, when there is an update or there is the management of new resources, the data management plan can be pre-fill or update directly. The LifeWatch Italia also provide a data portal for the management and sharing of fair dataset and associated metadata. In the LifeWatch Italy data portal, user can share a single dataset, or they can create, customize, and share a kind of sub-portal where the researcher that are involved in a particular project can gather all the data set related with the same project or initiative. In LifeWatch Italy data portal, the description of the data is done using the ecological metadata language 2.2.0 version. This is a metadata schema in use in different other infrastructure, like for example GBIF. And it refers to different section, and all of these section contain different attributes. One of the customization that we done in LifeWatch Italy data portal is the integration of attribute of the metadata schema with semantic annotation. What mean that some attributes of the ecological metadata language are annotated semantically using concept or property from ontologies or control vocabularies that are deposited in EcoPortal. That is the LifeWatch ERIC repository for semantic artifact in the domain, in the ecological domain. Another functionality in LifeWatch Italy metadata portal is that the validation of the metadata. What mean that the XML file is can be checked against the EML schema validator, and if there are some problems, some issues, the user have a kind of alerts in order to improve the metadata. So it's possible also to have a data validation, in particular if the dataset that are uploaded contain information about taxonomy. This dataset can have taxonomic check. In particular, the taxonomic check is done using two different taxonomic backbone. One is the Italian taxonomic backbone, which consists of checklists provided by Italian taxonomists, and the other one is the global taxonomic backbone that is based on Worms, Catalog of Lives, and Workflow Online, that are the three more used global database for the validation of taxonomical information. So how works this validation? After uploaded the dataset and click on taxonomic check, the user need to provide the delimiters for row and column. After he can select domain or the environment and the taxonomic backbone to be used for the text. At the end, the system provides different score, both for Italian taxonomic backbone and global backbone. And the user has the possibility to have different action in order to decide if modify eventually the name, the scientific name used in his or her dataset, or harmonize this information with the global taxonomic backbone. So after the dataset and metadata are published, it's possible to search this dataset by different search interface. One is based on simple search by text, another one on map, and after we have advanced search that is based on some filter that are the attribute of the metadata schema in use in LifeWatch Italy data portal, and also is possible to search by portal. So when users select a dataset, can see more details related with the dataset, but in the end can decide if download metadata and data and in which format. In fact, thanks to also the semantic annotation, we provide not only XML of EML and the CSV, but we provide also JSON, a JSON linked data format that is very good for interoperability of data. Another platform useful in research lifecycle is related with the, in particular with the development of code. Data labs, in fact, give the possibility to user to create, to manage script in different language like R, MATLAB and Python, but also to produce from these scripts some services with structured interface and new data. In this case, the first step is create the project, so to create the new code, and the user need to provide description about this new code that are the attributes of a metadata schema. After the user can select and input data not only from local but also taking data directly by the data portal, because there is a connection between data portal and data labs. After the code is created and developed in collaborative way in interface of Jupyter Lab, and at the end, the researcher, the user can decide to publish the project not only in data labs, but also can publish metadata related with scripts and services directly in metadata catalog, or in alternative also the data in data portal of LifeWatch Italy. So at the end, we have also the metadata catalog that is a kind of main access point for different resources, not only what I show in the previous slide, but also research site resources, audio, workflow, virtual research environment, and so on. So what is possible to do in ecological metadata catalog is possible to add or import a new record, to also to harvest more records by API, both not only LifeWatch Italia system, but also other by other research infrastructure. For each resource, we provide a metadata schema. For example, for script, we have a metadata profile based or customized using the ISO 19139. And for each metadata template are provided information about mandatory field and also the possibility to select recommended value directly by list and also by controlled vocabulary. At the end of the process, the publication of metadata is possible only if the metadata is validated by validators, and the user can also require the assignment of a DOI. all the resources and society metadata that are published in metadata catalog can be searched by different web interface, simple search, and also with map and with advanced search based on different filters. When a user selects a particular metadata or resources, can not only see what are the different details, information provided by the metadata template, but also in LifeWatch Italy metadata catalog is also possible to have a fairness assessment. This is possible because the LifeWatch Italy metadata catalog was integrated with the Fuji tool. This is a web services that can programmatically assess the fairness of different digital objects using metrics produced by Fair2Fair project. This is very useful because not only for user but also for the validator of the system, because in terms of user, the user can see different score for each of FAIR principle and can improve the metadata schema provided, or in terms of editors, this kind of tool and also the assessment of the fairness can help the validator in order to understand if validate the metadata and also to assign a DOI to the particular metadata record. So thank you. If you have any kind of question, this is my address.
Thank you so much, Ilaria, for this very interesting presentation of the facility that can be offered by LifeWatch Italy and the technology that is behind. I don't know if we have questions at the moment.
I don't see any question in the chat. Congratulations for your talk.
Thank you.
So I'm going to give the floor to Guillaume and Sophie.
Okay, so thank you. With Guillaume, we are going to introduce the activities of French BIF, which is more or less the activities of our department, research department and public policy department too, which is called Patrinat. And we are going to introduce data standards and especially Darwin Core that we use within the GBIF network, but also for Biodiversa Plus activities. So sorry in advance if you already know Darwin Core. This is going to be a really introductory presentation, but I will try to speed a bit so Guillaume can show you the Biodiversa Plus implementation of Darwin Core. So I'm going to start with a really short introduction to GBIF, just so we have a bit more of context with the use of Darwin Core. GBIF stands for Global Biodiversity Information Facility, and it's an intergovernmental program and data infrastructure that was created in 2001 by the OECD Scientific Committee. And its objectives are to promote and facilitate the free and open access to biodiversity data. So I'm not going to focus in detail on the numbers that change every day, but roughly we are dealing with almost 3 billion data and occurrence data, and that are shared by more than 142 countries and more than 2,000 data publishers. So you can imagine that it's a really important volume of data, and these data can be really different before standardization. So these data come from different sources, such as museums, agencies and ministries, data that are collected through field protocols, field science, citizen science. We, in the last few years, even have data coming more and more from the private sector business activities, from universities, of course. So you can see it's a really diverse sources of data, and we really need protocols and rules to in order to share them and make them interoperable, and that's in order to match with the FAIR principles that were presented earlier. So GBIF put in place several recommendations and in the last few years in order to achieve that, such as open licenses and of course data standards that are used since the beginning to share the data and make them interoperable through different tools such as the IPT, but we will focus on standards today. So for a brief introduction to data standards, or even just standards, standardization, as was said by Edward Deming, does not mean that we should all wear the same clothes or eat the same thing, the same food, or live in the same building. But it just means that it's best if we use the same rules and regulations in order to adapt the standards to our needs. So it's exactly the same way to deal with data. We can use several standards and we can make them adapt to our needs for sharing data, but it's in no way an obligation to share exactly the same data with exactly the same fields and so on. It's just a way to facilitate the sharing of data. So as I said, a standard is just an agreed way of doing something. It's you can sometimes hear about conventions, restrictions, requirements, rules, norms, specifications. All of these can be defined as standards. And we use standards in our everyday lives, such as units of measurements, alphabets, even the smileys or emojis, codes such as the Morse code, the Braille code, numeral systems, languages, informatics languages, but also human languages, postal addressing, and so on. So a standard, in its definition, can allow you to restrict and make more easy to share the types of data, encoding schemas, formats, character encoding. So that's what really makes the data that you share understandable by other persons. For example, for character encoding, you might have noticed if you use a language such as French, or I think maybe Spanish or Italian, we often have accents on characters, and it can be misinterpreted by other languages, informatic systems such as English. So you will have like black boxes instead of an accented letter or special characters. So that's a problem with character encoding, and most standards should really take that into account and implement rules to fix this kind of issues. Standards can also be used for data transfer, so you can define application schemas, formats for your dataset structure, and even the way you're going to transfer your data to other databases or other computers, such as transfer protocols. So now to focus on biodiversity information standards, they're the rules that by which data are described and recorded, and we should use them in order to share, exchange, and understand the data, because it's the only way to make the data interoperable, as I said at the beginning. So there are several existing biodiversity information standards, such as the ecological metadata language that was mentioned earlier, the EML. So this one is a metadata sharing language. So it's usually not used for sharing data, but it is largely used for sharing metadata, that is to say descriptive information about the data. There's also other data for sharing standards, for sharing data, or multimedia information that can be linked to the data, such as the Audubon Media Description standard that is also known as Audubon Core. For genetic data, the GGBN produced its own standard, and other thematic standards exist, such as the standards for sharing ocean-related data. And for biodiversity in general, for occurrences and taxonomy information, we can use the Darwin Core. So what is Darwin Core? It's just a list of fields and their definitions related to biodiversity data. So I just want to add that Darwin Core is best for sharing information about biodiversity. It's not really a data managing standard. Of course, you can use it if you wish so, but it's a standard that is that has quite a large scope in order to take many different sources of biodiversity data into account. So it's best for sharing the data, and it might not be the best standard or the best way to manage your data when you're still working on it. If you want to know more about Darwin Core, you can follow the links at the bottom of the slide. We will share the slide after, but I'm not going into much detail because I think maybe Rui and other presenters might talk more about Darwin Core in general later. So just to continue with Darwin Core, it can be divided into field classes about record and dataset information, occurrences information, of course. That's one of the main set of fields for Darwin Core. Organism information, material sample, event, location, geological context for paleontological data, and identification and taxon classes. And if you need, there's also auxiliary classes such as resource relationship or measurement of fact that allow you to share variables and information about how different specimens or individuals relate to each other. So once again, if you want to know more about Darwin Core, you can follow the Darwin Core quick reference guide that lists all the fields, their definitions and examples, and it is already divided into classes. So at GBIF and GBIF France in particular, because I'm from the GBIF France node, sorry, I completely forgot to introduce myself. I'm the data manager for GBIF France. So I, in my day-to-day work, I help people sharing their data using mainly the Darwin Core standard and EML standard for metadata. So we don't have a specific way of using Darwin Core at GBIF France, but we have other uses that Guillaume will show you later. So that's why I'm just presenting the Darwin Core standard, because we use it as it is for sharing occurrence and taxonomical data to GBIF and event-based data too. So that's why I'm only presenting the main Darwin Core terms here, but if you have any questions, you can ask them in the chat or contact us at the email address that we put at the end of the presentation. So the main, not the main terms, but some terms, Darwin Core terms that are really important within the GBIF network are, for example, basis of record that allow you to describe the type of data that you're sharing within the GBIF network, but not only. So you can use different controlled vocabulary values such as preserved specimen, fossil specimen, living specimen, and so on. It's one of the only few terms in Darwin Core that has a controlled vocabulary, so you cannot put anything other than the values that are listed here. And it's used by GBIF to filter the data and to refine the requests that you can do on gbif.org. Then we have occurrence ID. So this is a really important field to fill in. You cannot share data within GBIF or other networks without a unique identifier for each and every one of your occurrences or taxon. So occurrence ID is for occurrences, and we have the equivalent for taxon, that is taxon ID. And another one is event ID for sampling event information that you want to share. So this is in this field you should put the unique identifier for your information. And it's mandatory. You cannot publish anything without it. So it's a really important field to share. And then we have other terms. So I just put here one example, but there are many other terms that you can use. These terms, country and country code, are quite important too, because that's what allow you to share the location of your occurrences or taxon information. I mean, there's also other geographic terms such as decimal latitude, decimal longitude, but if you share the coordinates, you should also share at least the country code, that is the ISO code with 2 letters for the territory, in order to check that the coordinates are within the right country or territory. And then if you have other information to share after using the main Darwin Core terms in the core classes, you can use extensions to Darwin Core, such as the Audubon media description, measurement of facts extension, identification history, and many more extensions that exist and that you can find following the URL at the end of this slide. For example, at GBIF France, we are dealing in the last few months. We've had a lot of requests to share eDNA data to GBIF, and we are following the GBIF guide for publishing DNA derived data, and this guide recommends to use one extension to share this kind of information. So I really, if you want to share data within GBIF or with a network that uses Darwin Core, I really recommend you to check the extensions, because there probably will be one that matches your needs. So I'm going a bit quick to let Guillaume continue after. This slide only shows you the place of Darwin Core, if I can say, within the global biodiversity data standards landscape. Darwin Core does not exist alone in a vacuum. It's linked to several other data sharing standards and metadata sharing standards. So you can see here that it can be linked to genomic standards, taxonomic checklists, paleontology standards, other standards such as ABCD that was presented earlier, that is more focused on collection data. Dublin Core for some terms that are comprised into Darwin Core, and so on. So it doesn't exist alone, and it's a collaborative standards that can be amended and improved by the community as a whole. So I really invite you to check the existing working group on Darwin Core terms, and to, if you want, suggest new terms or extensions. And just to finish, I'm showing you the contents of the Darwin Core archive. It can be useful to understand how it works. Darwin Core is a standard, but in order to use it, within the GBIF network, we use some tools, mainly the IPT, but not only that. IPT stands for Integrated Publishing Toolkit, and it's one of the tools that is used to transform your data sets into Darwin Core archives that are harvested by GBIF afterwards. So a Darwin Core archive is only a zipped file, a zipped folder that contains several files. So one of them contains the data. In this example, it's occurrence.txt. And then it also contains the metadata using the EML metadata standard. Because as I said, it's not possible to share data within GBIF without associated metadata. And the other file, meta.xml, is the file that is used to make the mapping. That is to say sorry, I'm losing my words. That's used to make the correspondence between the original files, the original structure of your data set, and the Darwin Core terms. So within the same Darwin Core archives, you have both the information, the data that you want to share, the metadata, and the mapping file. So the Darwin Core archive is encoded as UTF-8 to avoid special character issues, like I said earlier. And that's what is harvested by GBIF and by associated networks to share biodiversity information data. And I'm finished with my part of the presentation. Oops, sorry. I just wanted to show the French National Data Workflow.
Okay, if you can going too close, it's going to be great because it's already a bit long. We don't want to shrink.
Okay.
But then
okay, so
I think that Guillaume will need a bit of time then to...
Yeah.
So.
Okay, sorry about that. So I'm just going to pass this slide, and I let Guillaume show you tell you about the biodiversity use of Darwin Core.
So I will just give you an example of how I apply Darwin Core in monitoring of biodiversity, because that's my job, and it comes from experience from for the EFSA a few years back to share data about wildlife monitoring in Europe, but also epidemiology use case. And here I took one example from the Water Framework Directive, which is the electrofishing protocol. I will be quick, but at the end of the presentation, you have links with some papers, some reports that re-explain everything, not with this use case, but with others, and you will be able to go deeper. So in monitoring, we have often one protocol really well organized. Here, electrofishing, we go in a river, we collect some fish, and some of them we are measuring information, and we are not going anywhere, etc. So we do not have just occurrence, we have more information that we want to share. And we often end up with this kind of data table. So here we put all the column below each other. So we have 40 columns with a lot of precise information, of course, all in French, and that's what we have to standardize. So there will be a part in English, but I just keep it. It's quite obvious. The first thing we will have to do is to think about our protocol. And as scientists, it's something which is quite interesting when we enter the standardization framework, is to think about our protocol. So here we first have a station in which we have sampling points, in which we have fishing operation, we still have a sample operation, and we catch some fish. That's the batch, and some of those fish we collect information. And there is a structure in that, and that we can use it in Darwin Core. So we can describe our monitoring in different steps, and we can right now associate each column to each different step because they are organized. And in a flat format, we lose this organization. So here we find it back, and it makes sense usually. And now we are entering into the Darwin Core, and we will use most often just a few tables. The main one, the core one, is the event core, where we will describe the sampling events, mostly everything that we more or less can fill in before going to the field. And then the observation we will make in the field will fit in the occurrence extension for observation. There is another table that is very useful and that I split in two is a measurement of fact extension, where we can really describe information about either measurement, which will have a unit, and facts that could be color or anything like if it's sunny, yes or no, or a lot of information like that. And this table is made just of three information, could be more, but that three main: the type, the value, and the unit. And we also have for the associated to the event, the Naouel Core that can bring more terms to describe your own protocol. So we will then associate each core and extension to our information. And here, station, sampling points, operation, and sampling events are events. And then the fish lot and the individual measure of fish are occurrences. And we can see that events are nested in each other. And then also what we propose is that occurrence can be nested in each other. In a fish lot, you can pick a few fish and have measure of them. And that's an occurrence, a few fish, in the fish lot, which is another occurrence. And here you associate also the measurement of fact where you need. So you can describe and organize your data sets in a more standardized format, graphical metadata, where you will have all your information that you will find in your different table. And now you can start to work really with the Darwin Core terms and to find in the Darwin Core the right terms that match your own columns, your own information, and maybe add some new information because they are useful when sharing your data to other people. So Darwin Core is not only about matching term to terms, it's also to organize your data in a different way, in a structured way. So it ends up in tables, of course, not just in graphics, in tables with different terms that you will find different terms for the different level of your events and the associated measurement of fact. And the same goes for the occurrence with the measurement. And the same goes for the individual measurements, so the sub-occurrences with their measurement of facts. And here, to simplify because Darwin Core does not control the vocabulary, most often, we are starting to use some simple rules that should reduce the diversity of what we enter. We have not applied them here, but we propose that everything to be to avoid capital letters, everything in English, everything in singular, and the units to be fully described. So here it's just mm for millimeters. It will be better to write millimeters in full and gram for other. Otherwise, there may be confusing elements. So I show you one example. Here is a graphical representation of another protocol, eDNA, for instance. That's another example with citizen science bird counts, and we can describe it also in Darwin Core. Here is another example where we describe results of a monitoring. So the estimated value of a density of roe deer in one area, and we use also the nesting of measurement of fact to have confidence interval, etc. So here is another example of important database in Europe, the Sigma database about epidemiological case studies in Europe. So we were able to match it with Darwin Core. So to summarize it, about the difficulties we have and the advice. First, we have to acknowledge that it's working. It works. We were able to fit most of our use case into Darwin Core, and that's a really interesting point, that we can put everything in Darwin Core, and it's hard to find use cases that are not matching with Darwin Core. It requested to do that a few elements that are not yet present in official Darwin Core, like the recursivity in occurrences and measurements, but the GBIF is working on that. With the new model should allow it. We need a few more terms. The event type could help us to describe if we are in a station, in a sampling operation, etc., to really know what we're talking about. The humble core is quite new and will likely need to be precise or adjust in the way we are using it. There are still some limits in Darwin Core. If you have a habitat or ecosystem approach, it's not so well developed. Darwin Core remains taxon oriented, but that's a way to work. And if you have use case like that, it could be interesting to work with you to develop the Darwin Core. There is no element to describe non-biological observation. If you study marine mammals, sometimes you see a boat, and it could be interesting in monitoring to describe it, but it should not be so difficult. When you organize your events, you also have some issue to know if you put the location first and then the time or the sampling. There is no fixed rules, so do as it suits you the best. But it's often easier to follow the logic of the protocol, and the graphic metadata that I show, we cannot yet send them, but they are really interesting way to present your data, and the color code is also useful. And it will be the list. So here are some links about the documentation. The first one is my reference, my starting point, and then there is two documents that I publish. There is the Darwin Core terms, and we try to develop a Shiny app to help us to do the work and to deal with the data themselves. So take it, improve it, and send it back to the community. It will help. Thank you.
Thank you so much, Sophie. Thank you so much, Guillaume. I think that if Sophie is remaining with us, I will ask only at this stage if there are burning questions for Guillaume for the second part, and then we are going to have a discussion on the Darwin Core schema altogether at the end.
I can see a question that just arrived in the chat. One question, do you use one measurement of fact extension for each core, or more than one is allowed? Guillaume, I'm not sure. Yeah, because you use the extended measurement of fact.
So the measurement of facts is one table. Here I split it in two just because it's easier to have one extension linked to conceptually to the event and another one to the occurrence, but it could be the same table, the same text file. Currently in GBIF, you're not allowed to have measurement of fact linked to occurrence that are linked to events, but in OBIS they have implemented that, so it will be feasible in sometimes.
Actually, I think it's possible on the IPT, but I'm not sure how is it managed by gbif.org afterward.
But basically, it's just one table.
Yeah.
Okay. Okay. Thank you so much. Thank you so much, Guillaume.
Thank you.
So my suggestion is, if all of you agree, to have only 5-minute break, not more than 5, and then we can rejoin at 11:16. I see that we are still all here, and so I will give you the floor for your intervention of the Portuguese approach, the approach in Portugal that you have.
Thank you, Alberto, and thank you also for the invitation to be presenting our experience at the Portuguese knowledge BIF here in Portugal. I will share my screen. From the Portuguese perspective, also the use of the Darwin Core, and because the presentation by Sophie and Guillaume was so good, it will facilitate a lot, and possibly also I was to take less time in this presentation on how we use the Darwin Core in Portugal. Of course, we do this in scope of GBIF. You already know very well what is GBIF. This is the main outcome that we can see here. But in Portugal, we do this also integrated in the Portuguese research infrastructure for biodiversity data for Biota, which is the Portuguese participation to LifeWatch ERIC. So in GBIF, we can see data like this. The Asian Hornet, for example, has been some years ago a topic of concern in Portugal. It also created some social alarm. So in GBIF, we can do this very easy, which is follow the distribution changes of the species around Europe in this case. And the good thing about this is that this data is being confronted to a GBIF from many different sources, aggregating and creating a better map on the distribution. So this is another way of showing what Sophie was already presenting before. But the most interesting number here for me is the number of publishers, more than 2,000 publishers from all over the world. So these publishers are distributed in for 142 countries. And why this is interesting? Because we are dealing with many different institutions of many different missions with different objectives, different cultures, different ways of working. So if we have all this variability and difference in the ways they work, is that possible to then aggregate data in a way that if some institution here in Portugal is publishing data, we can have someone in Australia looking and using the data? That's really a social and cultural problem that I think it's interesting to analyze. And the use of the Darwin Core standard is a tool that we use for that. Sophie also showed this, the interesting part of this workflow that goes through GBIF from the sources of data to the final users is on the left different types of sources. Then we go to the standards and supporting open licenses that GBIF uses. But then in the middle, we have the infrastructure. And I would focus in this part for a bit. We have the integrated publishing toolkit, IPT, it was already mentioned. It's the tool that we use to provide data in the way that GBIF can integrate and ingest in the global infrastructure. And in the IPT, we provide, as Sophie said, the data in Darwin Core format as a CSV table or text table, but also the metadata. So the IPT also serves as a platform so that we integrate the metadata that is associated to data. That's what Sophie already said. So, but the good thing of IPT is that guides you through different forms to insert the metadata that then becomes a specific profile of the ecological metadata language. And then this needs to be integrated in the global infrastructure through an ingestion process. And that's where things are done very well, but can be more improved. Why? Because in this ingestion, not every information, not every column in our Darwin Core tables are fully ingested and especially indexed. What this means is that for the final user to search data, he can only search data to create filters on the indexed fields. GBIF has advanced a lot in the last few months and years on the capacity of indexing more fields, but this still creates some stress to the way that the then final users can access to data. in Portugal, what we do the starting point of that workflow. So we as a national node, we work with different institutions, helping them to mobilize, to organize their data. And that is done not only from technical support, but also through training and also infrastructure support. And the training here is very important. What are we talking about? We are talking about occurrence data, but also event-based data. So we have different dimensions of data. This is an example based on a picture of a mushroom taken in field. Only from this picture photograph and additional some information, I can see what was seen, when it was seen, by who, in which habitat conditions, and the date when it was seen. So to fully document these different dimensions of data, it's good to use some references. So for that, we can use several sources as authority files for the taxonomic data. We can use guest years to document properly the location. A list of collectors that is a reference for our collections or our institutions. We can use database checks to identify, for example, if the date that is in the dataset is correct or is wrong. And we can also use thesauri that were mentioned also today in the workshop to support the better description and standard description of the conditions where the data was done. So this then helps to organize our data in Darwin Core, and Darwin Core is a community-driven standard. So it's created by experts and researchers from all around the world that through the TADWIC organization agree on a common standard. And this aspect of having standards that are curated at community level through TADWIC and accepted from this community is very important because it supports that adoption of Darwin Core globally. Darwin Core, this was already shown by Sophie. It's a list of terms organized in classes, and for a specific class, Sophie showed this because we in GBIF use this all the time. So in fact, in our training workshops, this is one of the most targeted terms in the classes because we need to fully document each of our records through the basis of record. So we need to explain to the people that are preparing data to be published how to document each of your own records. And this is not easy because many times we ask our participants in the workshops, in the training workshops, bring your data that will be organized through the workshop. And the interpretation of, is this in human observation? Was this a machine observation? Was this an occurrence record? These differences in the interpretation of the record happens. So it's only through training and work that we can fine-tune this use of the Darwin Core. So the training is essential. We in Portugal and many of the national nodes of GBIF have regular training workshops. In Portugal, we do this through different kinds of workshops, but in all of them, Darwin Core is touched and used as an example. We train on data publication through GBIF. We train on data quality and data cleaning on the Biodiversity data context. We train on georeferencing, and we also train on the use of Specify, which is software for data management of biological questions. In all of this, we use Darwin Core as examples to the support of the data standards and as how can we better organize and use our data. One thing which is particular, and from our experience we have seen many times, is people are eager to adopt these standards and participate in the community. But sometimes with all this enthusiasm, they say, I'm going to prepare my database for my biological collection using Darwin Core. And I say, no, you should prepare your data using Darwin Core principles and guidance if it's possible, but there are other type of data that you need to have to manage properly your collection that Darwin Core doesn't support. So be aware of that. So find the best organization for the database that you are using to the management of the collection. And when we do that, have always in mind that then you will map that, you will make correspondences from these fields to the fields in Darwin Core. So that's the other important part. But don't use Darwin Core for the management, Darwin Core format for the management of the collection, because, for example, loans or gifts that you do in your collection will not be supported by Darwin Core. So that's something that only through training we can deal and solve with the users. And also then we extensively use also other tools provided by GBIF, like the data validator, then to check if the standardization of the data through the Darwin Core format was providing the right standard or the right way of formatting data. And we can validate that through the data validator. So in Darwin Core, in fact, Sophie mentioned a lot occurrences, but Guillaume mentioned datasets that are organized through an event core. And in GBIF, we can also have a third core, which is Taxa, which supports datasets with checklists. These are the three main. And the way these different types of datasets organized through the Darwin Core change depending on where you are. So globally in GBIF, we can see that almost 90% of datasets are occurrence-based, so the core is occurrences. But in Portugal, if we take the full number of, we have around 350 datasets published by Portugal. Most of them are not occurrence-based, are sampling event-based, which means that for supporting the needs of our publishers, we need to use the sampling event-based. And in Darwin Core, then, Darwin Core as used and interpreted by GBIF, it is used as a star schema. So we have a core table, it can be the event, the occurrence, or the taxon. And then attached to that, we can have extensions that give more flexibility to add more types of data to our dataset organized in Darwin Core archive that Sophie explained very well. But this star schema only can have one level. And this creates some constraints on if we have richer data. Imagine that, as Guillaume was mentioning, I have event-based data to which at the event level, and then I need to add more measurement of fact information. But then at the occurrence level, which is in that case already an extension of the core, which is in the event, then to the occurrence, I also need to add measurements of facts. GBIF doesn't support this. So this is creating some limitations that right now makes that some data that then is published through GBIF are poorer than could be in the scope of the data it is provided or it is served through GBIF. One way of overcoming this is, as I mentioned, the use of extensions, measurements of fact, or the other extension similar to this, extended measurements of facts, or Humboldt extension that Guillaume mentioned, is a way of adding more data and more different types of data to your dataset. It's kind of a generic class to support many different types of data, but then we also have dynamic properties, which is a term which is totally general in concept. So structuring your data as we see here in JSON format, for example, you can fully document everything you need. But this data then is not searchable through GBIF. So it gets somehow limited in the past. It's standardized, is made available through the IPT. It can be also accessible through gbif.org, but then it requires some work from the user to reinterpret these terms and use the data. So because of this constraint, GBIF and the whole community noticed that there is a need for development. So that's why GBIF is developing a new data model that I will very briefly introduce here using slides provided by GBIF. That was a recommendation of the 20-year review of GBIF. And the way to do that was to identify different use cases and provided by the community on which types of data or which types of sources of data need to be supported. So, for example, EDNA metabar coding, camera trap, tissue samples, etc. So these different use cases and more can be added. Served to check what would be a data model to support every type of data. So the way this process was conducted was first create a use case narrative, try to interpret that as a template, and check if that interpretation of your use case and description fits the conceptual model, and then adjust the conceptual model if needed for your specific case, and then organize that. So that has been the exercise developed by GBIF and partners in the last 2 years, I believe, which is providing ways of making sure that every type of data we have in the source they get constrained as it then is used by GBIF. So this is how the process develops from one specific use case with the metadata, with the database schema, then how, or the data model, then how this can be seen as rich information provided to the user. So this is another example of a use case for this case in this instance for camera traps. So what is done is from this data model, only part of it will be required for specific use cases. So this is to say that this is another example based on environmental DNA. And I will go back to the last slide in a bit, but just to explain that we will not require the users to understand this full data model. What we will require is the users to define their case use, and through the IPT with the adaptations that is being developed, then identifying the use case, it will be able, the IPT will be able to identify in the data model which fields are relevant for your use case and show that in a way that you will provide the data and the data will be not constrained or destructured as many times happens now. So these are the latest developments from GBIF, and the final mention to the importance of identifiers. This guide that was published through GBIF, it is already 13 years old. And this is one of the reasons, personally for me, it was difficult in the beginning to understand what is personal identifiers, why do we need that personal identifier, and this has been a process that luckily GBIF insisted from the very beginning, which ends up that now in an assessment of interoperability of the fairness of GBIF as a whole that was exercised through the project AGI ACE, the data space for GBIF as described was compliant in the 3 of the 4 fair principles. Only in interoperability we couldn't comply that same at 100%. So this is still a process to do, but most of this success is on the importance of persistent identifiers that I first started like 11 years ago. I didn't understand, but now I can understand well. So this is around a community and around GBIF would provide this the contribution in terms of fairness of the data that's provided. And thank you with this. I hope I didn't take too much time.
Thank you so much, Rui. It was really, really interesting and really great. So thank you so much for the presentation. If you agree, we are going to go on with the next one, and then we make a general discussion with all participants. So I will give the floor to Andrea Tarallo.
Yes.
For the LifeWatch Italy case or LifeWatch ERIC case.
Yeah, it will be mainly LifeWatch Italy.
Yeah.
Thank you for inviting me. My name is Andrea Tarallo, and I work for the Italian National Research Council. And in LifeWatch Italy, that, as Ilaria explained already, is the Italian node of LifeWatch ERIC. I will talk about our approach on how we use the Darwin Core to standardize the terminology of the data that we gather in LifeWatch Italy community. That is slightly different from the way that you already saw with the other presentation. So, well, this is briefly to present LifeWatch Italy. It is organized as a joint research unit of more than 30 partners in Italy. We have university, research centers, some also private partners that are mainly a data provider to LifeWatch Italy. But the digital infrastructure is actually maintained in Lecce, which is here in the south of Italy, at the University of Salento, where we have this fortunate joint collaboration between the seat of the CNR IRET, the University of Salento, and one of the seat of LifeWatch ERIC, that is the service center. So thanks to this tight collaboration, we can develop and maintain our digital infrastructure that Ilaria showed you already, at least some bits of it. This was already presented. So this is how the Darwin Core archive is organized. So it has this minimum of 3 files. So we have one required core files, which are the data in tabular format. Then you have the meta file, which provide the annotation of the variables that are within the data file with the Darwin Core terminology. And then you have resource metadata. I want to see if I have puntatore, yes. Okay. And you have the metadata which describe the data in the archive. It is already been said, this is a great way to mobilize, exchange data about occurrence, specimen occurrence in biodiversity research, especially because this archive is self-explaining. So people can exchange information just by exchanging this archive, and everything is inside the metadata that explain the data, and the annotation where humans can actually go and understand in the same way what does it mean that specific variable. This is a great way for this kind of data, but as a community we receive many different types of data. This is just few examples. I already showed you how huge is our Italian community. And so from our data providers, we do not only receive occurrence data that are, let's say, classical data that are managed through GBIF with Darwin Core, but we have also other kind of data like traits data, such as morphological traits, physiological traits, behaviors. We have even experimental data, and we have a lot of accompanying variable to the occurrence that are usually environmental parameters. We have huge community of marine biologists in LifeWatch Italy, for instance, and when they collect specimens, they also measure environmental parameters such as temperature, salinity, conductivity, and so on and so forth. But we have also here, for instance, geology, geomorphology related data. Recently, we are collaborating in a project that is mobilizing records from stegofauna that are zoological records from aquatic animals that lives in caves. So together with the presence of the species, we have also a description, for instance, of the morphology of the caves, or of the composition of the rocks and all these kind of elements. So when we start developing infrastructure in LifeWatch Italy, we soon realized the limitation of using the Darwin Core to manage those kind of data. Okay, some of them have been already discussed in previous presentation, such as the recursivity in occurrences and measurements, as Guillaume pointed out. And this for us is exacerbated by the fact that most of the time we'll have individual data, for instance, about the traits for single specimens. But for instance, also we would like to find a way to manage data in an extensible way, because we cannot a priori say what kind of variables we will have from our community. Okay, so we were seeking a system that is extensible and easy to use by our community, but on the same time that it also allows to be queried also in a complex way, not just among the data set that are mobilized by the community, but also within data sets, so at level of single observation. And this was achieved, I will show you later, by a semantic search. We built a semantic engine that make use of ontology to actually browse the data that are annotated by controlled terminology. So this is the how we approach this process. This is again already being shown by previous presentation, because the problem is always the same. We usually receive the data from the scientific community in formats that are not standard, are usually Excel file with the variables, the headers of the columns that are not described by standards, that are not controlled terminologies. So what we do is to transform this file, this Excel file from the researchers to a standard type of file that can be shared and can be understood by machine. This is maybe one aspect that has not been highlighted too much this morning. When we talk about fair and improving fairness, it's not just using terminologies that we can all as human understand in the same way. This is important, but this is just one part of the fairness, because fairness is mainly about machine readability and machine actionability. So a machine should be able to go through this data set that is described in a way that the machine can understand the meaning, so the semantic of the data. So basically, our strategy is to use the Darwin Core terminologies in combination with other terminologies, other standard terminologies, to map the columns of our files that we keep as a flat files. So we don't use the Darwin Core archive strategy, but we keep using flat files. But each of the variables are described, are mapped, or better say, are annotated with variables that comes from standard vocabularies. So we primarily use the Darwin Core, that is, of course, worldwide accepted, and this long-term managed by the TIDWIG group. But then we use also other terminologies to describe the let's say, the variables that are not managed by the Darwin Core. So we use, for instance, the thesauri that we develop in LifeWatch Italy. We have the eco portal, LifeWatch ERIC maintain eco portal, that is the repository for semantic artifacts. Ilaria showed it this morning. And we deposit in eco portal the thesauri that we build in-house. These are usually thesauri that came from bottom-up activities from our community needs that asked us to build specific thesauri in case there are not standard terminologies that can be used. But in eco portal are stored also terminologies that are developed by other communities that can be also other search infrastructures such as Alter, for instance. We use the vocabularies that are stored in the NERC vocabulary server. These are mainly used for the environmental variables or also the chemical variables, for instance. And we use also the terminologies that are stored in the repository of the Ontoport Alliance, and Naouel showed BioDiff portal, for instance, this morning, but there are others like BioPortal. We use the terminologies of all these standards to map and describe the data that we receive in order to have standards persistent identifier that are attached to the terminology and can be then understood by machines, as I was saying before. And this is one of the example of file that we receive, a practical case study. As I told you, we usually receive Excel files because this is not the standard, but apparently is the standard for researcher to use Excel. And we always have the same problem that have been already showed before in another presentation. So we have here, for instance, the application of headers that cannot be really managed by machines. We have the variables that are described in are described not by using terminologies that are standard, but terminologies that can be understood only by human in this case. Also the data that are within the variables are not standardized. Here, for instance, the taxonomy is adopt different way to be described. So our work is to support the researcher in harmonized, standardized this kind of data to be then mobilized to our data portal. And here you see the diversity of terminologies, of standards that we use to map our the variables that are used by researchers. So this is the same headers of the file I showed you before, with the original label on the left, the label that comes from the standard terminology here in the column label, and for each of them the terminology that we used to describe that single label. So this is basically the annotation of the original label that is provided by the researcher. And as you can see, we use primarily Darwin Core. Most of the terms are mapped by the Darwin Core. Here also the taxonomy has been extended to be more clear and structured in a standard way. But we also use other vocabularies and thesaurus. For instance, here we use the environmental thesaurus that is developed by the Alter community and is stored in the Eco Portal. This is to describe the metabolic rate, but we use also the zooplankton traits that have been developed by us, by LifeWatch Italy. And here in the blue, underlined in blue, you see there is the ModNet chemistry aggregate parameter names. That is one of the vocabulary that is stored in the NERC vocabulary server. So this process has been done by data stewards in LifeWatch Italy. So we have human resources that can support the researcher in doing this work manually. So this is requires a lot of time, because you need a back and forth with the researcher to be sure that the mapping is done in a correct way, and you need people that are dedicated to a single project to do it. But thanks to, this is the file that is finally created. But in the last 3 years, thanks to the LifeWatch Plus project, it is a national funded project by the Italian Ministry of Research, we were able to develop a huge infrastructure and to automatize this process. So with the new data portal that also Ilaria showed you this morning, we are able to annotate each of the variables from the file that the user is submitting to the portal with standard terminology. So that means that here you can see, you can introduce your file in tabular format, that is a CSV file. The system can read the header, the first line of the file, and for each of the original description of your file, it suggests you a label that comes from a standard terminology. Or you can decide to directly type in the URI, that is the system of the identifier, of the terminology that describe your variable in that column. So in this way, there is an automatic annotation of the variable, so that the data that are submitted to the data portal are automatically annotated and can be afterward be searched using the semantic annotation, the semantic platform at the observational level. Okay, so we can do complex queries on our data that are not just filter of the column of the variables, but we can ask, for instance, things like, give me all the observation that has been done between this and that longitude and latitude at that specific temperature by that specific data provider. and the system give you all the back all the observation. This for us was very important because the system, the data portal, it is connected with other platforms in LifeWatch Italy, such as, for instance, Data Labs that again Ilaria showed you this morning. And users of Data Labs can query directly the data portal using semantic and enriched queries to retrieve the single observation. So this is why we adopted this different way of managing data in our infrastructure. And this is to show you that the annotation, the structure and mapping of the files from Excel to CSV using standard terminology is just one part of a bigger pipeline that do not stop here, but go through checks, quality checks, especially the taxonomic check, and then the system requires the user that is submitting the data to compile metadata that means to the LifeWatch Italy metadata catalog and are mirrored on the LifeWatch ERIC metadata catalog. So together with the data set, we have also metadata in EML 2.2.0 that are connected to the data, to the data set. Along with the publication, afterwards, you have the export of the data in CSV, so you can download back the data in CSV, but also you have the JSON linked data. This is very important for interoperability. Ilaria already elaborated on this, but this is one important point because one of the advantages of this approach is that even if it's customized for our infrastructure, it provides data that are highly interoperable with other systems. And indeed, on our to-do list, there is already the project plan to build an in-house script to export the data directly in Darwin Core archive to be directly digested by GBIF, for instance. And this is possible because we have this kind of expert as JSON linked data. Other advantages is, as I told you, the integration with other platforms in LifeWatch Italy, especially the Semantic platform and Data Labs. This system allows the user to extend the file, the data file, as long as he or she wish. This is again to overcome one of the limitations of the Darwin Core that if you want to extend your table, you have to actually ask the terminologies to be extended with a new term, and this might be a long process. Instead, with our system, you can find the vocabulary, the terminology which describe your variable, and just put into the data table and annotate, and the system understand it. And as I told you, the system is highly interoperable with other systems. So this is all from my side. Thank you very much.
Thank you so much, Andrea. Thank you for this presentation and highlighting also the characteristic of the use of Darwin Core in LifeWatch Italy. So now I see there are a lot of comments in the chat. I don't know if there are questions because I didn't open, so I leave Sophie.
Yes, there's a question. I think this to Andrea. Do you do it only for data collection in Italy? And I mean, sport scientists for organizing their data to be uploaded in LifeWatch Italy?
Yeah, thank you. We started as a national-based project And the first approach was by gathering data from the Italian community, because we have several data providers. But then we started to be partners also in international projects, mainly European-based projects, in which we were asked to be partner to help to support the scientific community of those European projects to mobilize data to the LifeWatch Italy data portal. So quick answer is no, we don't do it just for the Italian community, even if this is our core activity, but we are happy to support also other activities worldwide.
Okay, thanks, Andrea. I'm also thanking the participant that has uploaded in the chat additional usable information. I'm looking something from Astrid with a guide on how to publish freshwater data on GBIF also. So it's really very, very high contribution level that we are receiving. So other question that you say in the chat for the speakers?
No, in the chat, not now, but if there are questions for this section, we can take them now.
Of course, we have time, and then the participant can raise your hand, and then the speaker will reply.
Actually, I can do a quick. There was our questionnaire. There was a question: how to manage simulated data? Can it be archived, entered into public workflow? So
okay, who want to reply to this question?
We can ask any of the
exactly.
Yes.
Asking to the speaker.
Yes. Any of the speakers has experience with simulated data?
I can answer from our side. We didn't have no exercise like this. Still, we've been working with real data, but I do see an interest in using simulated data, especially in this context that I was mentioning in the development of the new data model that GBIF is doing. So to stress the model with different types of data and different links that need to be maintained through the whole workflow process to make sure that it fits the necessary steps and results for the final user to be able to get data fully documented. So while yes, the use of simulated data we haven't done yet, but I see a lot of interest on that.
Thank you so much, Rui. I think there are a lot of data that could come from simulation, from modeling, and then could be surely usable. Additional question from the participants. You have seen that there are different, that Darwin Core is actually flexible, so that you can adapt Darwin Core to your needs, and then according to your needs, you can have different kind of solution that could be advantageous, and then you can use it. And then the other message that you got from the lecture, the speaker, is that it is always possible to have mapping between different standards. So it's there's the possibility to integrate data even if they are developed in ABCD or in other kind of standard, then it's always possible.
There is Naouel with the raised hand.
Okay, thanks, Naouel. And
Yes.
also Fran. Naouel first.
Okay. Yes, I have a question about Darwin Core and the connection or crosswalk with schema.org. Is there already some work in this direction? Do you plan, so if any of the speakers already plan such representing also the datasets in schema.org?
No, I'm sorry, I didn't listen to which version you want Darwin Core to be made with?
To schema.org.
Schema.org. I'm not aware of that. So if I'm not aware of that, means that at least from our side here in Portugal, we have not done any exercise of that. But the knowledge on what you see here and what is schema.org that we don't know, and do you see any difficulties in the mapping?
Actually, schema.org is very general. It's what Google will use to collect the data sets and make them available via the Google dataset search. And so basically, it doesn't have the details of other standards, but it has like a more general properties. But that's the only advantage is that you have more how to say reach because of the it can be found, like the data set will be found also in a Google data set.
Okay, great. I will look at something that was not mentioned about Darwin Core is that is an evolution of an existing standard for long, which is Darwin Core. So in fact, Darwin Core is reusing some of the terms of Darwin Core and keeping the concept as it was defined in the beginning. So I imagine this kind of exercise can be done also with other standards.
Mm-hmm. Okay.
Just to answer a bit on that, I did a quick research, and apparently it's been discussed by John Vixorek and Steve Baskoff, two of the creators of Darwin Core, on GitHub. It was just a suggestion at the time. It was last year, so I can send you the GitHub issue if you want to. But it's only a short discussion, but they're aware of that, and maybe they will work on that in the future.
Okay, thank you.
Thank you so much for the answer for the question. Then I see that Fran.
Hi. First of all, thank you very much for very, very thorough presentations, providing both, a smooth introduction to those not acquainted with the topics and also very advanced aspects for data standardization. I am based at Fondazione Montmartre, but I also am the co-chair of the Data Standards Working Group of the International Biological Society. So I'm used to work with tagged animals and have a lot of information about tagged animals. Apologies if maybe some questions have been raised by, for example, Luca De Lucchi or before or someone else, but I will interrupt it in my concentration on the presentation here. So my question is, how do you see the possibility to reconcile rich information about single individuals with the more classic observation of species, spatiotemporal observation of species? I know that there is a working group also on that. My colleague Peggy Newman for the Atlas of Living Australia, together with Peter Desmet for InfAI, have been great contributors. But so do you see this possibility coming? And then I stop. My question doesn't want to be so long, but for example, sometimes we have camera trapping studies, and at the same time we have marked animals in the same area. So would it be possible to see such information to be reconciled and not to say wasted? Thank you so much.
Thank you for the question, Fran. I don't know if we have answered from our lecturer, from our speaker.
Well, if I can, I think this is mainly for the GBIF community. From our side, this is one of the motivation that we use this different approach, because we have a lot of individual data that are very useful. So with a flat management of files, it is easier for us to manage those kind of data. So, but I leave the floor to the GBIF colleagues.
What I can say on this is that we try to keep all the granularity on that standard, use that we can. So going to the level of the organism level, it has been a struggle because many of the management of the data we do focus on the occurrence, it's not the organism, but the support for that is there in the Darwin Core, and GBIF keeps that information with the record. So at some point to being possible to use this to do this and links with other types of data that is linked to that. Although GBIF focus on the use of Darwin Core.
Thank you, Rui, and thank you, Andrea, for entering the discussion and giving some reply to the question and also comments about that. We are a bit late with time, so if there are not other questions, even because I think it's then useful to spend 5 minutes to try to have a wrap up and also to try to start figuring out a direction where we can take after this workshop, because the 2 days have been so relevant that I think that even though there is not any plan for a continuation at this stage, then probably it's going to be useful to make some step forward on this line. So if it is fine for you, Rui has already raised the point, then we need training, we need capacity building, and then that it has already been done in the Portuguese community. And then I will leave the floor to Cosimo Vallo from LifeWatch ERIC to discuss exactly this point. So the capacity building, developing skill and expertise, and creating community of practice in LifeWatch ERIC.
Okay, thank you, Rolf, and good morning, everyone. I'm Cosimo Vallo. I'm a training officer in LifeWatch ERIC, and actually, while I will be focusing, as Professor Basset mentioned, on tools and support for all the services that you have presented today and yesterday. And especially I am very happy and glad to be invited here to present the experience that we have in LifeWatch ERIC, both in terms of developing skills and knowledge and sharing knowledge, and in creating community of practice. I'm happy that Professor Basset recalled what Rui commented earlier on. I've seen also a couple of comments in the chats and one from, I can recall one from Astrid that mentioned guidelines. And basically everything that I've seen today is, in a very broad sense, somehow knowledge sharing and training. So I'm going to present to you what we have in place, and maybe if, as Prof. Basset mentioned, you decided to go for further developments of this workshop, maybe you can resort to. So here is, let's say, the training architecture. So everything that we have put in place in LifeWatch ERIC for our users and our communities. So following a bit what the title of the presentation is, I have let's say divided the our structure, our architecture in 2 parts. One part which is focused on training, learning, and education, and capacity building in general, which includes our training catalog and a training platform. And I will obviously elaborate more on the 2 just in a moment. The other part is more focused. I mean, obviously, this categorization is not really that, let's say, precise. I mean, it's obviously a mix of the two. It's just for ease of understanding and presentation. The other part is the user support and the community building. What we have developed is a community platform, which basically brings help, is a tool, a platform that helps to bring people together, and an help desk. So all these four tools and services that you see are our way at supporting our users and communities. For the services that LifeWatch, ERIC, the various national nodes, and the partner institution develop. So now moving into the actual elements. So the training catalog. The training catalog is basically a list of all the entries of the metadata of all the resources and training products that we have identified as useful for our users and our communities. So in this catalog, basically, you will be able to find resources which are internally produced, accompanying, let's say, some of the tools and services and web services and workflows that LifeWatch develops, as well as other products that are produced by, as I mentioned, external institutions or partners or communities, and are value for the biodiversity and ecosystem science community. So this catalog is aimed at ensuring and enhancing the fairness of our training offer. And this because the metadata that you find are aimed at facilitating basically both the search by the user of the product and the training product that is really needed for his to address his needs. And also for machine, let's say, interoperability and accessibility, this catalog exposes APIs and can be recalled from other by other platforms and be shared and visible also somewhere else. So fair training catalog to enhance sharing, discoverability, and accessibility of and reuse also of the resources that are hosted here. This catalog also allows for contributors. Let's say if you if you want to if you have a resource that you would want to host and you reckon is important to host on our training catalog, you can get in touch with us and we can create a new entry for the resource. Then the resource is hosted where it is initially hosted, but the visibility is enhanced through the LifeWatch ERIC training catalog. The training platform. The training platform is instead our own, let's say, repository of trainings. It hosts training and learning and education products that we develop in LifeWatch at large, ERIC and national nodes. As you can see, this is obviously what you are seeing here on the right hand of the page is a screenshot of the home page and the link to the actual platforms. I had planned initially to actually quickly navigate them, but in the interest of time, I will leave that to your curiosity. The links are there. It will be in the recording anyway, and in the end, I will give you our email contact in case you'd like to get in touch with us. What is important actually to show and to mention about the training platform in particular is the fact that, and you can see here, we have 5, let's say it's a simplified navigation where a user arrives and is, let's say, immediately guided into what sort of content is looking for. So, education and schools, the eCampus, which is courses, the manuals and tutorials for our VREs, services, and so on, and the webinars. And then there is this category is international projects. Basically, as LifeWatch ERIC, we participate into international projects, and what we in training VPs of international projects. And what we developed with the years, with the experience, is project-specific sections of our training platform. So what I can mention about this is that, let's say, as a possibility, we can develop a section dedicated to, I don't know, Biodiversa Plus or projects that the community here may reckon useful in the future, and have a dedicated space for hosting training contents. So, and I go back again to what Rui mentioned, there is a big need for training here. I'm pretty sure that there are, and Astrid mentioned guidelines hosted here and there. That is definitely good. What we found useful is giving to the users and the communities one single go-to place to find every training related to the Biodiversa Plus, in this case, community. So that is something that we reckon is useful and can be a possibility for this. Now, moving to the other aspect, moving to the community of practice building, and moving to the, let's say, user support in a more general and broad term, we have developed the community platform. The community platform is basically a space where users and members of the platform can create their own working group. They can meet, interact, and collaborate. It's a space for designed for enhancing collaboration, networking, and promotion of opportunities. So you see here working groups. There are already 12 working groups. There could be the possibility to create. There is the possibility to create as many working groups as you want, provided that you register and create the working group. In this working group, you can invite members, share documents, share links, launch discussions, and so on and so forth. Again, in the interest of time, up to your curiosity to navigate it. And the last element, which is a bit of, let's say, overarching somehow, is the help desk. The help desk is the place where, well, I mean, everyone knows what an help desk is, but this is basically our own help desk, and is designed to respond to specifically to the LifeWatch products. What is an added value, and what is an enhanced value of this help desk is that it's a living, let's say, tool, and the more our users and our community use it, the more it will grow and increase the knowledge base. Every ticket that it's opened is somehow made added to the long list here. So let's say you may join the help desk in order to put a new ticket, but then you discover that there is already the answer there. So this is again our way at providing all the support possible for our users. Well, this is the team. It's myself and Eleonora Parisi, both based in Lecce and fully concentrated and focused on the training action of LifeWatch ERIC. At this page at this link, on the LifeWatch portal, you can see read a bit about our approach to training and education. Just briefly, this is what we use to coordinate actions. We have a training working group with 14 training focal points now, with from all national nodes and the central office. So we meet monthly, we share information, we share. It's a two-way communication. They inform us about what is going on in their offices and nodes, and we provide them information on the general direction of the institution. This is very good because this allows us to populate the training platform and the training catalog that I showed you earlier, and therefore strengthening the position, the ERIC position, the LifeWatch ERIC position within our community and the scientific community of reference. And yeah, it's been very I ran through the slide, but this is it. If you have doubts or anything, you can go rewatch the recording, or just drop us an email at training@lifewatch. I mean, it's really hard to forget. training@lifewatch.eu, and we are more than welcome to respond to you. And with this, thank you very much.
Thank you so much, Cosimo, for this presentation. We allow us to have an overview of what can be supplied by LifeWatch ERIC for the community. There are certainly a number of material already useful for the bio monitor coordinator projects that are dealing with this aspect on the data portal, on the data catalog of LifeWatch. That is nice. So thank you so much. I don't know if there are questions that...
Yes, there's a question for Cosimo, and the question is, what is the license of material and who choose it?
Well, it's the let's say intellectual owner of the material. So we can put different licenses based on what the requirement is. Yeah. So it's the training catalog really only hosts the metadata, so that's really we can choose and select. That's if I'm not mistaken, the license field is actually an open field, so we the owner can actually fill at their preference.
And what about Cosimo? What has been produced by LifeWatch?
Same. It's CC BY, so LifeWatch needs to be mentioned.
Yeah, but it's then
credits must be given. That's
completely open.
Yes, completely open, absolutely. And also, like we have the possibility, like for our training platform, we have the possibility to restrict access, and we do that for example, schools and short programs that we have, just because there are sensitive data in it. But all the rest of our contents are fully open.
Okay, so thank you so much. Then, if there are not other questions, since we are late, and I see that we are still almost 50, which is absolutely great, because We are 20 minutes late, and I'm sure that people need to move to different meetings and different activities. So I would like, first of all, in order to avoid to forget something, I would like, first of all, to thank a lot all the speakers, all the organizer, and all participants. And in the organizer, I would like particularly to thank Sophie, Martina, and Senem, who have managed to solve a technical problem that can happen when we are online. We're reducing the online support and the ICT component. Can always happen. It shouldn't never happen if we were in presence. But then they managed to solve the problem in a very, very short term, and I thank them so much for this. Of course, was simply an unpredictable case. Then we have discussed a lot in terms of what interoperability, harmonization, interoperability, and the standard, and discuss a number of components of Darwin Core. What I would like to say, and I'm sure that I'm also, I don't know if Harry is still with us, but I'm sure that I also can speak on behalf of Harry for what has been done yesterday. We were not planning to do something more on that at this stage, but then due to the participation, the interest, the discussion, the contribution that we have received, there is something that I would like to say about the future, because we will remain available as the task 2.2 and the specifics of task dealing on the dataset and data harmonization and interoperability, available to give support to all the participants in order to deepen and to enter more in the practical use and implementation for Darwin Core in their own project. So feel free to be in contact with us. Feel free to ask us additional support, and I'm sure that also the participants to this workshop will help us if needed with their knowledge and expertise. But it's something that of course we will do. We are going to discuss inside WP2 of Biodiversa Plus, and we are going to bring in the next meeting that we will have, if we can figuring out a possibility to organize an additional event that could be an hands-on workshop, possibly in presence, when I think the project are going to completely in their first year, so they're going to have the data, they're going to have different needs, and then we will try to find out the condition, if it is possible, to have an additional workshop that could be in presence and online, in order to support the participant to make some step forward on practical activity. We have done as LifeWatch for different community and in different occasions, and so we will be happy to bring this in Biodiversa Plus as an additional, we can say, milestone, or an additional delivery, or if it is possible, we need to discuss, but then we will give you a feedback. And that, I think it's all from my side. I don't know if Harry is here and would like to say something.
Yes, I'm here. Yeah. Thanks.
So if you would like to say also something at the end of these 2 days, I'm going to leave you the floor.
Okay, thank you very much, Alberto. This was a very nice workshop, and I have actually learned a lot in this workshop by myself. And it was interesting to follow that we have gone into the much deeper of data management with the second part of this workshop, because the first day it was the basics, and it was just learning what we should do first, and now there are some practical things, and you have gone into the very, very deep parts of practical things here. So I am really happy about this part of the workshop. I would just like to also thank all the participants of our 2 days here, and Alberto and his team, and also, of course, all our members of working group who have been working with this workshop. And after this workshop, we will send the compiled presentations and also the recordings of the workshop. You will receive them with a small reminder to send the DMPs as well, updated DMPs. But I will send personally, at least from the day one. But I will discuss with our working group how we will compile also the second day here. So thanks.
Thank you so much. And then I think that with your words, Harry, we can close the 2 days. And thanking absolutely everyone for the participation. It's been really a pleasure for us to organize and to participate to this workshop. So thank you to all of you.